In the realm of plant genome editing, a seismic shift is underway, transforming the landscape with groundbreaking tools that promise unparalleled precision, versatility, and efficiency in genetic modifications. This evolution transcends traditional site-directed nucleases like ZFNs, TALENs, and CRISPR-Cas9, which have already revolutionized genome engineering by enabling targeted mutations and gene knockouts. But here's where it gets interesting. Emerging systems are pushing the boundaries beyond the conventional double-strand break (DSB) mediated methods, introducing a new era of genome and transcriptome manipulation. Advanced tools like LEAPR, SATI, Restore, Rescue, ARCUT, SPARDA, HACE, Type 4a CRISPR system, and transposon-based techniques such as TATSI and piggyback are opening up uncharted territories in genetic modification. Some of these technologies enable DSB-free DNA editing, precise base substitutions, and RNA editing without altering the genomic DNA. A monumental leap forward for regulatory compliance and organisms with intricate genomes or limited regenerative capabilities. For instance, LEAPR, Rescue, and Restore pioneer RNA editing, while SATI offers DSB-free DNA editing, and ARCUT boasts reduced off-target effects and cleaner DNA repair mechanisms. The Type 4a CRISPR system, on the other hand, facilitates gene silencing instead of editing. Transposon-based methods like TATSI and piggyback, along with helicase-based approaches like HACE, provide alternative pathways for genetic modification. SPARDA, utilizing the prokaryotic argonote protein as an endonuclease, adds another dimension to DNA editing. The transient and reversible nature of RNA editing tools, particularly Restore and LEAPR, introduces an epigenetics-like control mechanism in plants, potentially enabling tissue-specific and environmentally responsive trait expression. Meanwhile, innovations such as ARCUT and SPARDA employ chemical guidance for enhanced precision, decreasing dependency on biological nucleases and mitigating off-target risks. The modularity and programmability of these tools are revolutionizing gene function research, synthetic pathway design, and targeted trait stacking, positioning plant genome editing as a platform for crafting adaptive crops that can withstand environmental and nutritional challenges. Though many of these cutting-edge tools await application in plant systems, their potential to create climate-resilient crops is immense. This article, co-authored by experts from institutions across India and the United States, including the University of Delhi, Sharda University, Michigan Technological University, Gautam Buddha University, and YMCA University of Science and Technology, highlights the transformative impact of these technologies on agriculture. As we stand on the brink of a new era in plant genome editing, one thing is clear. The future of crop science will be shaped by the convergence of genome engineering and systems biology, heralding a new age of precision agriculture. But wait, there's more to this story. While the potential benefits are vast, the ethical and regulatory implications of these technologies cannot be ignored. As we unlock the power to engineer crops, we must also consider the long-term ecological impacts and the equitable distribution of these advancements. And this is the part most people miss. How will these tools change our relationship with nature and our understanding of what it means to cultivate food? We invite you to join the conversation and share your thoughts on the future of plant genome editing.